Hey everyone, this is Princy Saraf, student of BSc 6th SEM, pursuing my bachelor from Dr. Hari Singh Gaur University, Sagar. This is my mid-2 assignment. I would like to express my deep gratitude to Professor Dr. Dipali ma'am for giving me such an opportunity. I would also thanks for her advice and assistance in keeping my progress on schedule. So the topic of my presentation is Entamoeba Histolytica. Firstly, I would give the introduction. Entamoeba histolytica are pathogenic amoeba that are widely known for causing intestinal and extraintestinal infection in human beings. Entamoeba histolytica falls under the phylum Protozoa, the class Lobosia, and the order Amoebida. Lamble in 1859 first discovered the parasite, and Losch in 1875 proved its pathogenic nature. Entamoeba histolytica causes dysentery by invading the mucosa and submucosa layers of large intestine in human beings. Entamoeba histolytica are commonly found in tropical and subtropical countries. So, amoeba are of two types. First is free living and intestinal. Entamoeba histolytica is an intestinal amoeba. All intestinal amoeba are non-pathogenic except Entamoeba histolytica. It is infectious. All free-living amoeba are opportunistic pathogens, means when our body immunity is low, they attack to our body. So in this presentation, I would discuss about its morphology, life cycle, pathogenesis and clinical features, laboratory diagnosis, treatment and prevention. So, morphology is Entamoeba histolytica occurs in three forms. First is trophozoite, persist and cyst form. So firstly I will discuss about trophozoite. It is a vegetative or growing stage of the parasite. It only forms present in tissues. It is irregular in shape. The size ranges between 12 to 60 micrometer and average 20 micrometer. It is large and actively motile in freshly passed dysenteric stools, while smaller in convalescents and carrier. So in this diagram, it is the picture of Entamoeba histolytica showing ectoplasm and endoplasm, and inside the cytoplasm there is ingested erythrocytes and nucleus. So cytoplasm, outer ectoplasm is clear, transparent, refractile, and inner cytoplasm is finely granular, ground glass appearance with nucleus, food vacuole, erythrocyte, leukocyte, and tissue debris. So we know that pseudopodia is a finger-like projection formed by sudden jerky movements of ectoplasm in one direction, followed by the streaming in of the whole endoplasm. Typical amoeboid motility is a crawling or gliding. Pseudopodia formation and motility are inhibited at low temperature. Nucleus is spherical, 4 to 6 micrometer in size, contains central karyosome surrounded by a clear halo anchored to the nuclear membrane by fine radiating fibrils called the linen network, giving a cartwheel appearance. Trophozoites form acute dysenteric stool, often contain phagocytosed erythrocytes. Diagnostic feature: this is not found in any commensal intestinal amoeba. So these divided by fine binary fission in every 8 hours. So these are killed by drying, heat and chemical sterilization. Then the persistic stage. Trophozoid undergo encystment in the lumen. Before encystment, the trophozoid extrude its food vacuole and become round or oval 10 to 20 micrometer in size of persist. It contains a large glycogen vacuole and two chromatid bars. So in this picture it is showing that a large vacuole and two chromatid bars are present. First is mononucleate, second is binucleate, and third is quadrinucleate cyst. Then the cystic stage is spherical 10 to 20 micrometer, and there is three types of cyst: early cyst, binucleate cyst, and mature quadrinucleate cyst. So early cyst contains a single nucleus and two other structure, a mass of glycogen and 1 to 4 chromatid bodies or chromatidyl bars. As the cyst mature, glycogen mass and chromatidyl bars disappear and the nucleus undergo two successive mitotic division to form 2 and then 4 nuclei. So let us discuss about the life cycle of Entamoeba histolytica. So there is infective form, mature quadrinucleate cyst passed in feces of convalescents and carrier. Mode of transmission is men acquire infection by swallowing food and water contaminated with cyst. Cyst wall is resistant to gastric juice, so that is why it is not destroyed in our stomach. And cyst reaches the cecum or lower part of ileum due to alkaline medium. Cyst wall damaged by trypsin, leading to excistation. So this is the life cycle of Entamoeba histolytica. So in this diagram it is showing life cycle of Entamoeba histolytica begins and ends inside one single host, that is an individual human being. So first is cyst stage 1. Infection occur by ingestion of mature cyst through fecally contaminated water or food. Due to protection from walls, cysts survive several days and sometimes weeks. They are responsible for transmission. Stage 2 is excistation. Excistation is the process by which cysts transform into trophozoites. When the cysts enter the ileum of the small intestine of the host, the process of excistation begins. The trophozoites are released in the small intestine, and from here they migrate to the large intestine. Then the third stage is trophozoid stage. Trophozoites are unicellular parasites that measure from 14 to 18 mm in diameter. They multiply in the small intestine by binary fission to produce cysts that exist via human stool. Several trophozoites remain inside the lumen of small intestine. The rest attach themselves to the intestinal mucosa, enter the bloodstream, and further grow in the extraintestinal region of the host like lungs, liver, and brain. So this is the flowchart of life cycle of Entamoeba histolytica. So cyst in the contaminated food and or water, then it ingested by the human being, then it becomes a metacyst in a small intestine, metacystic trophozoites, and in colon it becomes trophozoites. Then persist and cyst 1 to 4 nuclei, and it passed through feces. Pathogenesis and clinical feature. Entamoeba histolytica causes intestinal and extraintestinal amoebiasis. Intestinal amoebiasis is pathogenesis. Lumen-dwelling amoeba do not cause any illness. They cause disease only when they invade the intestinal tissues. Not all the strains of Entamoeba histolytica are pathogenic or invasive. So differentiation between pathogenic and non-pathogenic strain can be made by susceptibility to complement mediated lysis, phagocytic activity by the use of genetic markers through monoclonal antibodies and zymodyne analysis. The metacystic trophozoite penetrate the columnar epithelial cells of crypts of liver cone. Penetration is facilitated by motility of the trophozoite and the tissue lytic enzyme histolysin. Mucosal penetration by amoeba produce discrete ulcer with pinhead center and raised edges. So ulcer appear initially on the mucosa as raised nodule with pointing edges, and they break down discharging brownish nectorial material contain large number of trophozoite. Ulcers generally do not extend deeper than submucosal layers. So this is the clinical features of intestinal amoebiasis. The incubation period is highly variable from 1 to 4 months. The clinical course is characterized by prolonged latency, relapses and intermission. The stool are large, foul smelling and brownish black, often with blood streaked mucus intermingled with feces. So this is the specimen showing amoebic ulcer in colon. So it is infectious with Entamoeba Histolytica virus. RBC in stools are clumped and reddish brown in color. The patient is usually afebrile and non-toxic. So this is the flowchart that is showing sites affected in amoebiasis. So first liver is infected, and then through the liver it affects lungs, diaphragm, skin, and liver affects pericardium, peritoneum, stomach, intestine and inferior vena cava. Then it goes to the genitals area, and through that it causes spleen, suprarenal kidney. Thank you for listening me.